Episode 1 made a blunt point: a molecule that binds its target perfectly, but gets destroyed by the liver in minutes, or is toxic, is useless. This episode is the deep dive on the stage that quietly kills the most candidates, and it's the one place where predicting failure early is worth more than predicting success. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity, the properties that decide whether a molecule that binds its target can actually function as a drug inside a living body. Absorption: does it get into the bloodstream, surviving the gut and crossing membranes? Distribution: does it reach the tissue where the target actually is? Metabolism: how fast does the body chemically break it down? Excretion: how is it cleared, and how quickly? And toxicity: does it harm the patient, whether on target or off target? Historically, a large share of clinical failures trace not to the drug missing its target, but to poor ADMET, bad pharmacokinetics, or unacceptable toxicity. Catching those before the clinic is where much of the money gets saved. This is the sharpest expression of the whole series' economic thesis. The goal here isn't only to make things faster, it's to fail the bad candidates early, when a failure costs you a computation, instead of failing them in a phase 2 or phase 3 trial, when a failure costs hundreds of millions of dollars. Sit with that, because it inverts the usual intuition. A model that reliably says this beautiful binder is toxic to the liver, drop it right now, is worth more than a model that finds you a slightly better binder. Why? Because it moves the failure from the single most expensive stage in the pipeline to the single cheapest one. How does AI actually predict this? ADMET prediction is largely a supervised learning problem. You train models on databases of molecules whose ADMET properties have been physically measured, and then you predict those same properties for new molecules from their structure alone. A couple of documented platforms, ADMET AI, which is open and published in Oxford Bioinformatics, predicts a broad panel of ADMET endpoints very quickly from structure. It's useful as a fast filter over large candidate sets. And Schrödinger offers kickprop for property prediction, plus a 2026 predictive toxicology solution aimed specifically at toxicity, sitting inside that same platform used across the top 20 pharma companies. Now here's the architectural point that really matters. ADMET prediction is most valuable when it lives inside the generative design loop from episode 8, as one of the constraints during the search, not as a gate you slap on afterward. Think about why. If your generator only optimizes for binding, and you only check ADMET at the very end, you'll have spent the entire search exploring the wrong region of chemical space, and then you reject almost everything you found. Folding ADMET in as one of the simultaneous objectives is exactly what makes multi-objective design produce molecules that are actually viable, not just molecules that are high affinity. And the honest caveat, which bites hard here. ADMET models inherit the episode 5 generalization gap especially acutely. They're trained on historical measured data, which overrepresents certain familiar chemical classes, so their reliability on genuinely novel chemistry is uncertain, and that's precisely the novel chemistry generative design is built to explore. Toxicity in particular is famously hard. Some toxicities are idiosyncratic, and they don't show up until large, diverse human populations are actually exposed. That is exactly why phase 3 trials still exist, and why toxicity can't simply be predicted away. The model moves the cheap failures early, it does not abolish the expensive ones.